Absolute configuration of sugars and amino acid are determined by DL system based on the absolute configuration of glyceraldehyde. D isomers are related to D-glyceraldehyde. On the other hand, L-stereoisomers are related to L-glyceraldehyde. Now, amino acids are classified into five main classes on the basis of polarity of R groups. So first class, that is non-polar aliphatic R groups. Non-polar aliphatic R groups. Alanine, valine, leucine, isoleucine, glycine, proline and methionine are belongs to non-polar aliphatic R group. Alanine, valine, leucine and isoleucine stabilizes protein structure by hydrophobic interaction. Glycine has small side chain and it does not contribute to hydrophobic interaction. The next important amino acid belongs to this group is methionine. Methionine is a sulfur containing amino acid. It has thioether group. Now, it is a very important amino acid, that is proline. The proline has some unique structure. The secondary amino group of proline remains in a rigid conformation, and this rigid conformation reduces the flexibility of the polypeptide chain that containing proline. So determination of absolute configurations of amino acid. Now determination absolute configuration of amino acid. So how we determine the absolute configuration of amino acid? So absolute configuration of sugars and amino acid are determined by so absolute configuration of sugars and amino acid are determined by DL system. So DL system based on absolute configuration of 3 carbon sugar, that is glyceraldehyde. So absolute configurations of sugars and amino acids are determined by DL system based on the absolute configuration of sugar glyceraldehyde, that is a 3 carbon sugar. So absolute configuration of L-stereoisomer. So amino acids have possible two stereoisomer, and these types of stereoisomer are called as enantiomer is, we already discussed it in the earlier videos. Please check out those videos. So is related to the L-glyceraldehyde. Is related to the L-glyceraldehyde and D-stereoisomer and D-stereoisomers are related to are related to now we draw the draw for structure of L-glyceraldehyde. So we draw first L-glyceraldehyde. So functional group is CHO and another is OH. These functional groups are important because absolute configuration are determined based on the position of the functional groups. Now this side H and here CH2OH. As you can see, the position of OH is left side. So C, this is a chiral carbon because these are 4 different groups are attached with the same carbon. That is why it is called chiral carbon. And the glyceraldehyde is a 3 carbon sugars, 1, 2, 3. So this is 3 carbon sugars. The functional group OH present on the left side of this chiral carbon. That is why it is called L-glyceraldehyde. On the other hand, the structure of D-glyceraldehyde. Now, D-glyceraldehyde. So, sorry, this is carbon C. And these are 2 functional groups. One is CHO, another is OH. That is hydroxyl. Oh, sorry. This is H, sorry. The hydroxyl, in case of D-glyceraldehyde, the hydroxyl group present on the right side. So hydroxyl group present on the right side and here CH2OH. This is the structure of D-glyceraldehyde. So in case of D-glyceraldehyde, the hydroxyl group present on the left side of the chiral carbon. Now, in case of, we take an example of a simple amino acid, that is alanine. Now we draw the structure of alanine. So this is the carbon. Now here COO minus, that is carboxyl group is present. Now here H, very important amino group, functional group, that is amino group, and below the plane CS3. So these dotted line means the carboxyl group and CS3 present below the plane of the paper. On the other hand, the solid lines means that H and this amino group present upper side of the plane of the paper. This is below side and this is upper side. Now, as you can see, the amino groups, this is the amino group, these amino groups present on the left side of this chiral carbon, or it is also called alpha carbon. So NH3 group present on the left side of the alpha carbon, or you can say this chiral carbon. That is why same L-alanine. So structure of this particular amino acid, as you can see, The hydroxyl group, in case of L-glyceraldehyde, the hydroxyl group present on the left side. And in case of L-alanine or any types of amino acids whose amino group or functional group present on the left side are called as L-alanine. This is the aldehyde and carboxyl groups, both functional group present on the upper side, and that is why it is called L-alanine. On the other hand, the structure of D-alanine, like now we draw the structure of D-alanine. D-alanine, that is C double O minus carboxyl group. Here amino, functional amino group. Now here H, and below the plane of the paper, that is CH3, and this configuration is called a D-alanine or D-alanine. So this is the structures, and through this process we can easily determine the absolute configurations of amino acids.